And our final presentation this morning before the break, and it will be entitled High Throughput Techniques to Estimate Leaf Wilting in Peanuts. Our presenter is Sayantan Sarkar. Excellent.
Hello, everyone. My name is Sayantan, and the topic of my presentation is high throughput techniques to estimate leaf wilting in peanuts. My talk would include a short justification of my study, followed by hypothesis, objectives, materials and methods, result, conclusion, and future research. First, justification. There are various ways that peanuts are affected by low moisture stress. First of all, low moisture stress has a negative effect on the stomatal conductance and photosynthesis of peanuts. It results in reduced flowering and peg development. Moisture stress also lowers the nitrogen fixation in peanuts and cause aflatoxin contamination in peanut kernels. Therefore, low moisture stress is detrimental to peanut pod yield and quality. Now, the next question is why leaf wilting is important. It is important because leaf wilting is the visual symptom of low moisture stress in peanuts. It is caused due to reduced turgor pressure in the leaf cells, causing them to lose structural integrity. Therefore, higher the stress, severe is the wilting. The picture here shows how wilting progresses with severity of drought. The pictures on the left is one of the less wilted plant, and one at the right end is one of the severely wilted plants. Therefore, we can assume that low moisture stress can be estimated by quantifying leaf wilting. But can we measure it? Yes, leaf wilting can be visually rated based on the degree of wilt observed. Here, the scale used shows how to assign a number to leaf wilting based on severity. The least wilted plants get a 0, and the most wilted or physiologically dead plant receives a 5. However, visual rating is time consuming and is subject to human error. Is there an alternative method? Yes, remote estimation of leaf wilting based on variation in RGB or digital images of plants is possible. Here we can see on the left is a digital image of healthy plant, and on the right is a wilted plant. The healthy plant has the ventral or the upper surface exposed, causing it to be greener, whereas the wilted plant has the dorsal or the back part exposed, causing it to be yellowish and dull green. In the healthy plant, only the leaves are visible, whereas in the wilted plant, the stem and ground is also visible. These differences in images can be quantified. Based on that, I hypothesized that estimation of leaf wilting using RGB picture-derived vegetation indices is possible. And I designed a study with the objective to determine if leaf wilting in different peanut genotypes can be estimated from proximally and aerially taken RGB images. Moving on to materials and methods. This study was located at the Tidewater Air Wreck in Suffolk, Virginia, and it was planted in May 13, 2018. Treatments included 28 accessions from the US Minicore collection. The reason as to why these accessions were used, let me give a small background on their importance. The US peanut germplasm collection has about 7,432 accessions. For feasibility of phenotypic study, a core collection of peanuts consisting of 831 accessions representing the diversity of the original accessions were developed. In addition, an even smaller subset representing the genetic diversity for genotypic study and other extensive studies was developed. This subset was called the Minicore collection, including 112 accessions of the major botanical peanut types. We selected 28 accessions of the Minicore collection with contrasting traits to study for low moisture tolerance in the Tidewater region of Virginia. Each of these had 3 replications planted in randomized complete block design. To induce drought, rain shelters were used. Here in the picture, we can see the 3 rain shelters on the left. The red boxes on the right picture are the actual experimental blocks. The image is taken aerially, and the color contrast in the plots is due to wilted plots caused by induced moisture stress. This is the color contrast that I would try to quantify. For ground data collection, visual wilting rating on a 0 to 5 scale was used. The least wilted plant got a 0, and the most wilted plant, or the physiologically dead one, received a 5. Visual wilting rating was collected every 2 weeks, starting from 6 weeks after planting till physiological maturity of the plants. This represents the timeline of the plots that during the time it remained covered. The rainout shelter was pulled on the plots at 8 weeks after planting, and it remained for the next 7 weeks. For image data or for image collection, 2 methods were used. Proximal imagery using handheld digital camera. The images were taken twice at 3 weeks and at 5 weeks after covering the plots. For aerial imagery, a digital camera mounted on a drone was used. The images were taken on the day after the shelter was removed, which was 7 weeks after covering. For image and statistical analysis, the tagging and orthomosaic of aerial images was done in Pix4D. The aerial image cropping was done in ArcMap. The extraction of RGB color indices was done in BreedPix. The statistical analysis was done in SAS using core and reg statement. Here in both of these images, the left one is a proximal image taken using the handheld device, and the right one is a plot taken using aerial imagery. Both of them are of the same plot, and this is the difference we found when we compare a proximal image with an aerial image. Moving on to the RGB color indices derived. The first one is intensity. It measures the grayness of the image on a scale of 0 to 1 in a hue saturation index or a HSI color space. Second one is hue. It is color judgment based on the position in the HSI color space from 0 to 360 degrees. 0 degree is red, 60 degree is yellow, 120 degree is green, 240 degree is blue. These degrees are important because I would again mention them in my results. Saturation, it measures dilution of pure color or hue with white light in HSI color space. It ranges from 0 to 1. Lightness, it is the light reflected by a non-luminous body such as a plant. It ranges from 0 to 100. A index, it measures the shift from green to red in a CIE-LAB color space. B index, it measures the color shift from blue to yellow in a CIE-LAB color space. U index, it measures the color shift from green to red in a CIE-LUV color space. V index, it measures the color shift from blue to yellow in a CIE-LUV color space. Green area, it's basically the percentage of pixels within the 60 degrees to 120 degrees hue angle in CIE-LAB. Greener area is a subset of green area, which measures the percentage of pixels in 80 degrees to 120 degrees of hue angle in CIE-LAB. Crop senescence index is derived by the arithmetical combination of green area and greener area. Moving on to results. Here is the table of the R values correlating indices with leaf wilting. We can see here that hue, A index, U index, green area, and greener area had good correlation for both proximal and aerial data. The yellow values show the non-significant values. These are the graphs depicting the correlation of hue, A index, green area, and greener area with blue color showing the aerial data and orange showing the proximal data. The top left graph shows that the hue had negative correlation with leaf wilting, and the top right graph shows that the A index having a positive correlation with leaf wilting. The bottom graphs show that the green area and greener area have negative correlation with wilting for both proximal and aerial data. This graph tells a lot about the color change in leaves due to low moisture stress. Lowering of hue with higher wilting shows that there was a shift towards yellow hue from green hue, thus reducing the hue angle. Similarly, in the graph with A index shows that the shift from green towards yellow or from negative values to positive values with increasing wilting. Thus, a change in leaf color or a shift from green to yellow with higher stress is consistent in both these graphs. Similarly, a negative correlation of green area and greener area with leaf wilting explains that higher wilting, there is a reduction in green pixels in the leaves, which is a sign of stress. I used these indices to derive or to train regression models. The model with proximal images had an R-squared value of 0.56, meaning that it could predict 56% of the wilting, whereas the aerial images had an R-squared value of 0.8, meaning it could predict 48% of the wilting. So with this study, we can conclude that wilting can be estimated using RGB imagery. Proximal imagery is slightly better than aerial imagery and can be used for smaller plots or smaller studies, whereas the aerial imagery could be used for studies with larger number of experimental units or at a grower's field. In future research, first I would like to validate the model using a different study, the regression model that I got. I'm also trying to estimate wilting using near infrared and shortwave infrared imagery. One of my major goals from this study is to figure out a way to prepare an irrigation schedule based on remote estimation of leaf wilting, and to do further make it an automatic step, making it a progress towards precision agriculture. With this, I would like to thank Dr. Maria Balota for her guidance in this study, my committee members for their valuable feedback, my lab members and technicians for helping me with flights and data collection, and funding agency Virginia Crop Improvement Association and AFRI for funding my study. Thank you, and I would take questions.
All right, thank you for this presentation, and we have time for questions. The first question comes, are you able to differentiate different types of wilt, for example, wilt caused by drought stress versus disease, or is at this point a wilted plant a wilted plant?
So the question is, am I able to differentiate wilting caused by drought stress from diseases? Well, in this study, I did not include the disease study, and they were sprayed overall so that the diseases don't develop on the plants. So as of now, I'm not able to answer that question because I basically don't know if I'm able to do that. But my plants did not have a lot of diseases when they were wilted. So so yeah, that's the thing.
Okay, it may help with scouting for diseases in the future. The next question comes from Dr. Corley Holbrook. What genotypes do you think are most drought tolerant?
So the question was, what genotypes do I think are most drought tolerant? So in these conditions, especially in the tidewater region of Virginia, the runner types and the Virginia type of botanical types are the most drought tolerant ones, and the Spanish and Valencia did not do very good. On them, Win had the highest yield, so I'm guessing, well, Win had the highest yield in both the drought and in well-watered conditions. So I'm guessing that would be the drought tolerant one. However, I need to further evaluate and check my results.
Okay, thank you. And the final question comes from Dr. Dylan Wan. Do you think it will be possible to refine the RGB model enough to determine quantitative value estimates of wilting, or would it be best for just qualitative selection? Do you think you'll be able to refine it enough for quantitative value estimates, or will it simply be qualitative selection?
Yes, I think I would be able to refine it. In fact, I'm trying to include near infrared with the RGB so as to get a better get better results. And we know that leaf wilting has to do a lot with the canopy temperature, and that's why I'm also trying to include the shortwave infrared or the thermal imagery. So maybe including all of them could refine the results, but yes, I would definitely look into that.
Okay, this concludes the time we have for your presentation. Thank you very much.